Only. Hello guys and welcome back to Excel Share Academy. So today we are going to do the last topic of DNA repair. Okay. So in this, like the mutability and in the DNA repair, as I said, last what we are going to talk about is translation DNA synthesis. So this means what? It is allowing the DNA replication when there is a damage. Okay. So as I told you before also, sometimes you do have 10 checkpoints before you proceed to the next step. But again, what if all the 10 checkpoints are not working? Okay, so then the cell should decide whether the cell should undergo, suppose the DNA is damaged. Now, based on the probability, like whether it can repair in the next time, okay, the cell will make its own calculation and it will decide that either I can go for apoptosis when the cell understand that the repair is beyond, okay, beyond repair, the cell damage is beyond repair, then it will decide to undergo apoptosis. But if it says that no, maybe in the next round I can repair this thing, then it is going to continue the replication with the DNA damage. Okay? Again, the same analogy that let me have the broken DNA, but let me survive. Okay? So same thing, like being alive is more important than with having broken hands and legs. वही वाला same analogy. तो अभी अब cell क्या decide कर रहा है? It has decided that I'll continue to replicate, to divide, even though there is a huge DNA damage. Okay, so that is the cost now the cell is paying. So in many of the examples we have considered so far, damage to the DNA is mended by excision followed by resynthesis using an undamaged template, right? हमने जो भी अभी तक देखा, चाहे even if it is a mismatch base or nucleotide excision repair, every time we were using undamaged template. Now, but such repair systems do not operate with complete efficiency, and sometimes replicating DNA polymerase encounters a lesion, such as pyrimidine dimer ho gaya, apyogenic site, which is not repaired. So we know that pyrimidine dimer can be repaired. Okay, so Shruti, can you tell me what, by which method the pyrimidine dimer can be repaired? Akshaya, you? We have already discussed how we can repair the pyrimidine dimer.
Photoreactivation, right?
Yes, correct. So that is one method. Okay, it is also known as direct reversal. So you have got the enzyme called DNA photolase. And apurinic site, in the case of apurinic site, it is your NER method. Simple. So please do remember, it is important. So because such lesions are obstacles to the progression of the DNA polymerase, their replication machinery must attempt to copy across the lesion or to be forced to cease replicating. So what are the two options? Either it replicate, okay, across the lesion, यानी कि lesion को cross करके, जो भी hurdles है, जो भी obstacle है, it is crossing that, or it is forced to stop the replication. Now, even if cells cannot repair these lesions, there is a fail-safe mechanism which allows the replication machinery to bypass the damage or tolerate the DNA damage. And that is what we are going to discuss now. What is that mechanism? Having the translation damage. Okay, so you can see over here. So you have got the DNA damage is there. If it is repaired, then it's fine, no problem. Or the second scenario is that the cell is going to tolerate the DNA damage and continue the translation synthesis. So of course, you're going to have one strand undamaged, the another one carrying the damage, right? So this damage, this lesion is still present in the genome until a DNA repair pathway or mechanism comes and correct the damage. Clear? So it has not been repaired. Okay, it has not been repaired, but the replication has continued over here. Now, translation synthesis is catalyzed by special class of DNA polymerase which synthesize DNA directly across the site of the damage. Okay, so can you see the proteins over here? What we are going to see is, see, now whenever there is a lesion which is coming in the template during the replication, DNA pol 3. So of course we are talking about prokaryotes. right? If I would have said Paul, delta or epsilon, then it would have been eukaryotes. So first is, let's see in the case of the prokaryotes. So DNA Pol 3, okay. Now you can see a pyrimidine dimer is there. Can you see? So pyrimidine dimer is there, because of which there is a distortion in the template strand, okay. Now what will happen? The polymerase 3 will leave its beta clamp. Guys, can you remember the beta clamp? Remember the clamp loader? If you remember the figure and the video also, the clamp loader was there. It had 3 tau proteins, to which your 3 sliding clamp was there, and your DNA polymerase were there. So 2 for the lagging strand and 1 for the leading strand. Are you able to recollect from the previous lectures? And this
Yes,
beta clamp, okay, so this beta clamp, we know that what is the main function? It keeps your DNA polymerase 3 onto the template. It does not, it prevents its dissociation from the strand. So what will happen? It makes sure that it continues the replication, okay. Now you can see that since there is a distortion, okay, the damage is there, and because of this damage, the beta clamp and the DNA Pol 3 are getting dissociated, okay. They are getting dissociated because of the, now these will be, okay, so this beta clamp and the DNA polymerase are now getting replaced by the translation DNA polymerase, okay. So they are also known as Pol 4, Pol 5, DIN B, UMI, D2C. D2 means there are 2 subunits. Okay. So these are your translation DNA polymerase. Now they can extend the DNA replication across the thymine dimer. So you can see what had happened. So TT hai yahan pe, they have added GA. What do you think? What they have done? So what does the translation DNA polymerase has done? This one, this DNA pol. This is your translation DNA polymerase. So once it crosses the lesion, again your normal beta clamp and polymerase 3 will be recruited and they will carry out the normal replication. Okay. So what does the translation DNA polymerase 3 has done? If you see that TT was there. So on the strand, it was TT. So on the new replicating strand, it should be daughter strand. It should be A and A. But if you see here, they have added G and A. Okay. So what does it mean? So translation DNA polymerase, they add randomly without looking at the template strand. Okay. Clear? Now what is the next step? That in the next round, this should get repair. So we know that there are methods, there are checkpoints are there where the pyrimidine dimer can be repaired and the mismatch can be repaired. See, पहले अगर आपका pyrimidine dimer अगर है, if this is broken down with the help of the DNA polymerase, now what is the next step? To remove the mismatch. And we know that we have got the mismatch repair mechanism as well in order to remove the mismatch which has occurred during the replication. Guys, are you able to follow me? Should I repeat?
Yes,
Huh? Everyone able to follow? Tanvi, you have joined little late, but are you able to follow?
Yes, ma'am.
Okay, cool. Now, so in E. coli, DNA Pol 4 or 5 performs the translation synthesis. DIN B, UMU C. Okay, they are the members of the distinct family of DNA polymerase, which is found in many organisms known as Y family of DNA polymerase. ये इतना याद करने की जरूरत नहीं है, but you need to remember this sentence. Okay, the first one. Okay. Now there are 5 translation polymerase known in humans, 4 of which belong to the Y family. So this is right now not required. But if you in future, if you're working bioinformatics kind of work, if you're working, then you might need to know what kind of different polymerases are coming into which family tree and all. So right now with exam point of view, only first sentence is enough, that if there is a damage which has not been repaired, then the last step is translation DNA synthesis. Translation DNA synthesis means keeping the damage, tolerating the damage, and continuing the DNA replication. Okay. Yeah. So it's an important feature of these polymerases is that although they are template dependent, they incorporate nucleotides in a manner that is independent of base pairing. So randomly उन्होंने add कर दिया है, ठीक है? So this explains how the enzymes can synthesize DNA over a lesion on the template strand. Okay, because it's not reading the template, it is not reading the sequence, and hence it can add randomly. Consider the case of apurinic or apyrimidine site in which the lesion contains no base specific information. See, first case was where you had a thymine-thymine dimer. Okay, so even though there was the presence of base, still a random nucleotide was added. Okay, this is your first scenario. Now, what is the second scenario? That there is no base itself. Okay, no base. GATC में से कुछ भी नहीं है, nothing is there. Now, how it is going to do? So they are going to insert nucleotides again, which is not guided by the base pairing. So again, it is just going to add the nucleotides randomly. Okay, so these are the two scenarios by which a translation DNA synthesis can tolerate the DNA damage and continue the DNA replication. Okay, for example, a human member of the Y family of translation polymerase, that is DNA pol, ये H नहीं है, okay, it is called, I don't know how to make also. ऐसा कुछ, I forgot what do you call this. Anyone better in letters? So you have got alpha, beta, gamma, delta, epsilon, then kappa. This is, I think, neta. Neta.
Eta, ma'am. Neta
Neta.
or eta, ma'am?
I think neta. I don't know. Eta is also there. Let's check. So there is a table, right? Okay. Okay, I'm not getting it. Yes, yes. Oh, okay. It is eta. Correct. It is eta, eta. Eta. Yeah. Which belongs to Y family. Okay, okay. Good. So this is one example where you have got the eta, DNA polymerase eta belonging to the Y family. And you can just remember Y family means they all are represented for your translation DNA synthesis. Okay? So it is going to correctly add 2A residues opposite to a thymine dimer. That is good. So what is happening in the next, whenever there is a repair, so normally DNA photolysase will come and just breaks the DNA thymine dimer, and eventually you're going to have a normal DNA strand. Okay. Now, structural studies have showed that active sites of DNA pol eta is better at accommodating thymine dimer than is the active site of the another translation DNA polymerase, for example, kappa. इसका मतलब क्या? So, whenever there is a thymine dimer, we do have many different translation DNA polymerases. Okay, but if it is eta, then it is going to be beneficial because it can repair the damage, right? Instead of this kappa. Q, kappa will add random nucleotides, but eta will add 2A residues against thymine dimer. So, which is going to give the correct base pairing, right? Once it has been repaired. Guys, able to follow? Should I repeat? Chandni, is it clear to you?
Can you repeat it once again?
Okay, so so I was saying that, so we have got two scenarios, right? First scenario where we have got thymine dimer, second scenario where you don't have base, okay? No base scenario. Now in prokaryotes, we have got the DNA polymerase, which can, which can repair, which cannot, where these translation DNA polymerases are there, tolerating the DNA damage and continuing the DNA replication. Okay, so there we have talked about DIN-B, that is polymerase 4 and polymerase 5, correct? Now, in the case of human, so eukaryotes I'm talking about, we have got DNA pol eta. So we have got eta over here and kappa over here. Okay, so what I'm saying is that if suppose the organism is having thymine dimer, if eta as your translation DNA polymerase comes, okay, it can add randomly, not randomly, sorry, it can add two specific A residues against your thymine dimer, correct? So in the next round, when the DNA photolyase can come, it will make sure that the thymine dimer is broken, and now I have got TA base pairing, which is normal, correct? Now, in the case of kappa, if suppose translation DNA polymerase kappa is coming, so whenever there is a thymine dimer, it can add GA. Okay, that is, let us say that it is random. So next round of replication, photolyse is coming. It is making sure that T, G and TA is there. But it is not correct. It is not normal. Right? This is wrong base pairing. So in order to correct this, we have got the other like mismatch repair, mismatch base pairing repair is there. So another round of repairing is required. So it is always better that eta comes instead of kappa whenever there is a thymine dimer. Now, how we understand this? By the structural studies. So when you say structural studies, it is obviously the crystal studies, the protein crystal studies, using your X-ray diffraction. Okay, is this much clear? Then only I can proceed further. Yes?
Yes, ma'am. It was.
Does that mean that eta will always put admin base pairs only, or does that mean that in case of thymine dimer, it's likely to put admin?
It is likely because of its active site. So that is what I was telling. From the structural studies, they came to know about this. So let's see the structure.
So it is random.
It is random.
But then in case of that particular case, it tends to put A.
Yes. Okay. Yes. Just because the active site is meant like that. Okay.
Okay.
So if you see pol eta and kappa over here now, just imagine you have got the fingers and all, everything, thymine dimer is there. So what they have shown is that the active site, you know, the of eta, it always favors the DATP. Okay. Now the thing is that you need to see the structure alignment also. Can you make out the difference between these two active sites of eta and kappa? Anyone? Can you make out? Already it is written. So a thymine dimer fits well in the active sites of eta, allowing the polymerase to correctly insert two A's across the dimer. Now, can you see what is the problem in the kappa, why it is not possible for it to accommodate thymine dimer? Because of dash. Can you see in the figure? This is like find the difference in the figure. If you have played that game. Yes, anyone? Akshaya, Tanvi, can you see the difference in the two?
There is a structure known as M135.
Ma'am, it is in the structure.
Yes, what is that M135? अब तक तो मालूम होना चाहिए. What that M135 is meaning? What do you mean by M135 in a protein structure? Huh? Is it what? I'm waiting. Okay, I'll give you the hint. Amino acid. Now? Now? I've already given the hint. Which amino acid does M represents? Yes? No one? Oh my God. Akshaya? Which amino acid is it? Anyone?
Yes.
No one is able to remember? Huh? Yes, Akshaya? It
I
is?
forgot.
Oh my God. This you cannot forget. Okay, I'm telling you guys, this is really very important if you really want to be in the bio field. This is like, you know, till our death, we don't forget ABCD. In the same way, you can't forget this. So M stands for methionine. Okay. So when it is said that it is 135, 135 is the position number. This is the 135th amino acid in the protein structure of your DNA polymerase kappa. So it is because of the structural difference, the methionine, it is coming as the what to say, the hydrophobic, right? It is hydrophobic in nature. So it is coming out, and it is not because of its side chain, it is not allowing the thymine dimer to sit over there. Okay, and hence kappa has got, DNA polymerase kappa has got less chance of repairing thymine dimer as compared to your eta, polymerase eta. Okay, so we have got translation polymerase which favor particular kinds of damage. So maybe there are certain other kinds of damage where the kappa is responsible more. Okay, now with the help of the structural information, now can you guys think of it? Okay, in the next time. So this is how you get to understand. Yeah, see, it is easy. Okay, it is not that easy to crystallize protein crystals, because I have done and I failed very badly. It took 3 years for me to crystallize, but I couldn't get the crystal. It is a different story. But we should have the idea like which amino acid, okay, which amino acid is present over there. Looking at the structure, since here they have mentioned that it is M135. Okay, this is DATP. This they have written TT dimer, but in the software, in the where you are working, there it won't be written. Okay, what is, whether TT hai ki CT hai wahan pe. Looking at the structure, looking at the stick ball model, you should be able to understand what kind of base or what kind of DNTP or which amino acid is present. So that's why right now for the exam point of view, you don't have to remember the structure, but once you go later in the research or in the teaching profession, then looking at the structure, you should know that this is methionine or this is alanine or this is glycine. Okay, chalo, resting later. So guys, got the point? Please, huh? So because of its high error rate, translation synthesis can be considered a system of last resort. So it enables cell to survive even though it has got the catastrophic block in the replication, but the price it has paid. Okay. So the genes encoding the translation polymerases are expressed as part of the SOS response. What is SOS? Save our soul response. Okay. So damage leads to the proteolytic destruction of the transcriptional repressor, the LEX-A repressor, that controls the expression of the genes which are involved in the SOS response. So including those for DIN B, C, D, and inactive precursor of IMU-D dash. So these are nothing but your translation DNA polymerases in the prokaryotes. Okay. So the same pathway is also responsible for the proteolytic conversion of D to D dash. So ये सब हम detail में जाने की जरूरत नहीं है. They don't ask much. So cleavage of both LEX-A and IMU-D is stimulated by a protein called REC-A, which is activated by single strand DNA resulting from DNA damage. So what you need to remember is that whenever there is a single stranded DNA damage, okay, this gives a signal to the proteins so that they can leave the genes which are repressed. Okay. So what will happen when those genes are, when those proteins are removed from the there? So now genes are open for transcription. Okay. Now what kind of genes are getting transcribed over here, which is responsible for your Reg A? Okay. Now this rec A is going to activate your lex A and you move D, which will convert to D dash later. Clear? Now, this is responsible for the activation of the translation DNA polymerases, which is going to help in the translation DNA synthesis. Guys, clear है? और confusion है? Tanvi? Shruti?
I'm processing.
अच्छा. Shruti?
Yeah, it's clear actually. Yeah.
It's clear, right? The thing is that you need to just remember the previous repair mechanism also. So once you're done with this everything, okay, I'll try to upload as soon as possible. So once you're done with all the repair mechanism, then you'll get an idea कि ये अगर नहीं हुआ है, then the next. If not the next, then the third, fourth, fifth. Okay, but then you need to remember also if there is single strand DNA damage, so which one? Double stranded, so which one? If there is only a base missing, then which one? Okay. So like this, you need to remember or make a chart. See, understanding is easy. Only problem is with the remembrance. And now once if you can't remember, then this, then problem क्या होता है कि next part जो आएगा, next topic जो आएगा, it will be difficult to understand. Later we are going to talk about the transcription, translation. Everything is related to your application then. The protein, okay? So same way here. Now we next address the question of how a translation polymerase gains access to the stalled replication machinery at the site of DNA damage. तो अभी हमने कैसे देखा? When there was a stall, okay, because of the presence of the thymine dimer, the DNA polymerase 3 got stuck over there, right? Then only the polymerase 4 and 5 came in order to continue the translation DNA synthesis. Now let's talk about in the case of the mammals. वो हमने prokaryotes में पढ़ा. Now what about in the case of the eukaryotes? So in the eukaryotes, you have got your beta clamp, right? Beta clamp, which is associated with the DNA polymerase 3. Now, in case of eukaryotes, it is known as pCNA. If you remember pCNA, जो beta clamp का काम है, वो eukaryotes में pCNA करता है, ठीक है? So how this can be achieved? The sliding clamp, which is known as pCNA in eukaryotes, anchors the replicative polymerase to the DNA template. So there will be a chemical modification of the covalent attachment to the sliding clamp with a ubiquitin. And we know that whenever any protein is tagged with ubiquitin, it is going for ubiquitination. It means it is going for protein degradation by the proteasome. Clear? वहां पे क्या हुआ था? In the case of prokaryotes, it just got dissociated, right? In the case of eukaryotes, they are targeted for protein degradation by having a covalent attachment of ubiquitin molecule. Clear?
Yes, ma'am.
Huh? Okay. So its use in triggering translation synthesis adds to the growing list of the cellular process, which are governed by the tagging proteins with the ubiquitin peptide. So now we know once it is ubiquitinated, the sliding clamp recruits a translation polymerase which contains domains that recognize and bind to the ubiquitin. So this translation polymerase will displace the replicated polymerase and extends the site of the DNA damage. So क्या होगा अभी यहाँ पे? If it is eta or kappa, they will come and they will remove your epsilon and theta and they will continue the damage. Clear? I'll try to draw and explain. See, what is happening is that in the case of eukaryotes, so we have got polymerase delta and polymerase epsilon. एक leading और एक lagging में जा रहा है. Okay. Now we have seen that there is a lesion, which is not being repaired by the before described mechanisms. So now what will happen is that the DNA polymerase epsilon gets stuck over here. Okay. What we do is then इसका जो sliding clamp होगा, okay, इसके ऊपर आपका DNA polymerase है. So this pCNA, okay, this sliding clamp is known as pCNA. This gets tagged by the ubiquitin molecule. Okay. Now once this is tagged, this will be absorbed or screened by your translation DNA polymerase. For example, let's talk about ETA itself. So this translation DNA polymerase will displace this entire pCNA ubiquitinated DNA polymerase epsilon. Okay. So what will happen now? Abhi inke jagah pe your translation DNA polymerase ETA is there, which will just add and proceed. Okay. It will just add randomly A and it will proceed. Later again, your polymerase epsilon will come and continue. Next होता है जहाँ पे आपका photolyase or similar, these proteins are coming and removing the thymine dimer. Clear hai guys? And of course, which is tagged with a ubiquitin, they will go for protein degradation. Once it is tagged by ubiquitin, it means it is going for complete degradation. And this happens in the cytoplasm by the proteasome. Okay. Is this much clear?
Ma'am.
Huh? Same thing. We have only 1 minute, but I think much is not there. I'll try to finish in this time. So we have got the replicative DNA polymerase, the pCNA, the clamp is getting tagged by the ubiquitin. So this will be displaced by the translation DNA polymerase. Translation DNA polymerase will go and continue the synthesis, DNA synthesis. Okay. The damage is still present in the genome. Again, the replicative DNA polymerase, that is your epsilon or gamma will, epsilon or theta will come and continue. Later in the next step, the thymine dimer will be removed. That is the hope. Okay. Yeah. So, so we know that ubiquitination of the sliding clamp is a distress signal, and it is going to, yeah, same चीज जो भी हमने discuss किया. It recruits translation polymerase to rescue the replication machinery. In addition to a polymerase switching mechanism, data support that translation synthesis also use a mechanism of gap filling. Okay, this and all is not required. Yeah. 1 second, guys. Okay, so in addition to polymerase switching mechanism, there are certain data which also says that translation synthesis also uses mechanism of gap filling. So what does it mean? So if you see over here, so this much is clear that we have got the translation replicative DNA polymerase, the pCNA gets tagged by the ubiquitin. So what it says is that there are certain data which says that instead of getting stuck over here, what it does, it just skips it. Okay. It is just going to skip and come over here and start the replication. But there is a gap. This is also another damage. So this damage will be recognized by the translation DNA polymerase and it is going to fill the gap. Okay. And then of course, like Okazaki fragments, you have got the gap and that gap is filled by the ligase will come and it is going to seal the nick. Okay. So this is another method by which translation synthesis can happen. So whenever they have said that these are the models. So when we say that it is model, it means what? These are the two methods the scientists have proposed. And till now, we don't have the exact mechanism which says that either the first method or the second method is the best way or the real way by which the translation synthesis is happening in case of eukaryotes. Okay. So these are the two ways by which it does. Is this much clear? So instead of stalling the DNA, the polymerase is going to skip and jump and it will continue the DNA synthesis. Okay. And then the gap is filled by the your translation DNA polymerase and the nick jo hai, it will be sealed by the ligase. Okay. Yeah. So that's all. So from this, I can say that the DNA repair is over and from next time onwards, we'll be starting a new topic.